So Jörg Kuttler very nicely introduced calcium in a talk this morning, and he pointed out that calcium is important for pretty much every process that you could imagine in plants, which I totally agree with, and would just like to add that not only in plants, right now calcium is actively translating what you see and what you hear into action potentials that you can choose to ignore or process further. And anything that you remember from this talk, if you do, will be down to calcium, which is responsible also for short and long-term memory. So what I'd like to do today is talk you through some of the work that we've been doing on modeling spatial temporal dynamics of calcium. And the main driver for this is the biological problem, which I'll get to shortly. But there's a second driver, and that's the controversy that surrounds nuclear calcium spiking. So the field's about 20 years old or so. There have been a number of papers coming out. Many have been questioned in terms of their validity and correctness, and I'll get to the reason for that soon. And most papers in the area also mention the word controversy a lot, and that the gradients between these different compartments have been questioned. And the reason for this controversy is the structure of the nucleus. So this shows a high-resolution electron microscopy image of an animal nucleus. As you can see, it's covered with pores. And these pores are about 10 nanometers in diameter inside, possibly a bit more. And there's thought to be about 200 pores around the nucleus. So this is quite a massive cavity, and small to medium-sized proteins are thought to be able to diffuse through fairly freely. So you'd imagine that a small ion like calcium can zip through with almost no hindrance whatsoever. There are conflicting papers on publishing on the electrophysiology of the conductance of these pores, and that they could be in an open or closed state has been hypothesized. But looking at light and looking at this channel view, there's a huge cavity, so you'd imagine that calcium could diffuse through freely. If that's the case, it's difficult to imagine how you could establish a gradient between these different compartments. So it's interesting from that point of view. Just here's an interesting problem that modeling may help solve. But the main reason is the biology. So I'm aware that GARNet's mainly about Arabidopsis, and trustworthy sources tell me that that isn't Arabidopsis, that's a legume, Medicargo truncatula. To me, it looks like, well, I couldn't know the difference anyway. It's kind of green and planty. But legumes have quite a number of interesting features. So they can secure their own nutrient supply through symbiosis with bacteria. And this is a very fine-tuned communication process whereby the plants give off signal molecules, flavonoids, and these flavonoids induce in the bacteria all the machinery that's required to produce nod factors. And these nod factors are perceived by the root hairs, and they lead to these downstream effects that lead to these complex cell wall remodeling processes that lead to these nodules you see here. And those are bacteria populations that fix nitrogen for the plant. And they also form another symbiosis, which leads to quite a different outcome, but is quite similar in its nature. The plant again gives us signal molecules. These are perceived by the fungus, and this perceived signal molecule induces transcription of MIC factor, it's called here, that leads to these effects here. So it's a very interesting problem, how this communication takes place. And evolutionarily speaking, it's thought that the bottom one with fungus was important for the colonization of land plants. It's about 350 million years old, and possibly that the top one was derived from that. And that's indeed what the genetics tell us. A number of groups have dissected both of these pathways genetically, and there are quite a few shared components between these different pathways. So that's supporting this evolutionary angle on that. It also gives us hope that whereas a large number of plants have this symbiosis with the fungus, very few form the symbiosis with bacteria. But if the components are the same and we understood in sufficient detail how these signals are generated and how they're decoded, you could imagine that you could manipulate the system and trick plants that currently only form the mycorrhizae symbiosis to also perform the bacterial symbiosis. So I won't go into the detail of this pathway today, but just say that one striking feature of both of these pathways is this calcium signaling that seems to occur around the nucleus. And alternate pathways have been sought and not found to date. So it's tempting to think that calcium is the information transmitter. So that's an intriguing problem. How do you encode these specific differences of what's been perceived on the outside of the cell? How do you encode that robustly into calcium oscillations? How do you transmit these? And how do you decode them at the other end? And we're working on putting in place computational tools to study this whole pathway and the whole process. So one first thing we can do is we can try to build a model and see how these oscillations generated. So unlike Mike, we don't have any of the components and certainly none of the kinetic parameters, so it's quite a different situation. But we can hypothesize about what might be in the system. I'll get onto that soon. So we want to build a model of this system, and to model you have to make assumptions. We're going to make a few, I hope, fairly plausible assumptions. So it's known that hundreds of proteins in cells bind calcium. Binding kinetics is very rapid compared to the timescales we're looking at here. So all of these proteins for which we don't know the KDs, we're just going to exclude, and there are estimates that say about 99% of the calcium in the cell will be bound to other proteins. So we're just going to monitor the free calcium. So we know that they're bound, and then based on that, we're just going to say, okay, let's monitor just the free calcium. We've seen that these pores are absolutely huge compared to the size of calcium. So at least it's plausible to think that diffusion will occur freely between these two compartments. We just have to consider one, they're going to be in equilibrium. And they're plausible assumptions, but we'll revisit them during the talk. So how do we get oscillation? So again, we don't know the components, but something has to be letting the calcium in, something has to be pumping it out. So if you take a close look at a nuclear envelope, you see there's these big pumps here. And let's see if we can get that working. So 20, 40, 60, 80. So that's clearly in ATPase. Moving swiftly on. So we can hypothesize a number of different components, and we can describe these components by ordinary differential equations, and we can set up a model of the system, and we can fit this model, as been described in earlier talks, to experimental data. So we're just going to fit it to spike shapes. And we don't know these components, what these components are, but something has to be pumped in, something let the calcium in, something has to pump it out. We know the existence of an ion channel, DOMI1, this thing here in red, so we put that in as well. And then we're going to just see if we can get this thing to oscillate. So I should hasten to add that we can also get an oscillation using different components. We could have a purely ligand-gated channel. You could have a calcium-induced calcium release. It's not so critical what those components are, but this one has some nice features that I'll get back to. So we're going to solve the previous equations numerically, and what you're seeing here, just look at the fluxes. So we've kind of linked this to a model that's driven by the solutions of the equations. So the arrows are the ion fluxes, and you can see the charge changing too. And what you'll see is just that we can fairly nicely get the spike shape right. And if we just fit the spike shape in the model, the right frequency automatically drops out. So that's quite a nice feature. And of course, it predicts a number of electrophysiological features of the various components. One thing that's interesting too is that to get this to work, we have to, with this right spike shaping frequency, we have to set the Hill coefficient of this potassium channel to 2, and that fits quite nicely with a recent homology model that's predicted to have 2 calcium binding sites. So this model can do a lot. It oscillates, has roughly the right spike shape, right frequency, but it's limited. What it can't do, surprisingly, is initiate and terminate. This set of equations will oscillate forever. You can't stop them. And also, I told you that they have these two different symbioses, mycorrhizal and nodulation, and we can't easily change the frequency of this oscillation. Either it oscillates like this, or it doesn't, or the parameters just go completely off. So how can we extend this model to be more realistic? Well, we have to revisit the first assumption we made that we can neglect the buffers. So we put in buffers that describe the dye and some known proteins in the system. So we just add in the equations and solve them. So we thought they'd have no effect, but let's just add them in and see what the system's properties come out with. And if we put in the KDs of two known dyes, we see something quite astonishing, that the calcium buffers and dyes are buffers have quite a significant effect on the observations that we're getting. So every time you use a dye, you're perturbing the system. And this shows that putting in the KDs of the dyes, it's not a perfect match, but you can see that we're picking up something quite characteristic of the different dyes. And then we did something quite fun. So this in red here shows one of these stable oscillations. For those interested, that corresponds to a stable limit cycle in phase space. If you, to this existing system, just add some more buffer, what happens? So it will go in this phase plot. It will go through an unstable iterations and then settle into a different stable limit cycle. And if you look into the way this affects the spiking, you'll see it goes through this rapid spiking behavior. And that's something that's been described in the literature since about 10 years and been commented on. No one's offered an explanation for it. And it seems that if you just add more buffer to the system, you'd get this oscillation. So it's been observed with nod factor spiking. So that led us to an experiment with that and actually start off the oscillations with MIG factor induced and nod factor induced, and then just add one of the others. So the top two are mycorrhizae induced oscillations, and then we're adding nod factor. And you see you get this very rapid spiking before it sort of settles into a more stable oscillation. Likewise for the second one, they're just different concentrations. The below one is nod factor first, and then adding MIG factor, and again you see this different oscillation. So that's suggesting that upon perception of nod or MIG factor, there's actually being either released from the membrane or somewhere else in the cell. You're releasing a protein that can bind calcium buffer, into the nucleus. And this is actually kick-starting the oscillation. So we can show that buffers being released into the nucleus can initiate a system that initially wasn't spiking, can start the oscillations. You can cause this rapid spiking. You can change the shape and the period of the oscillations, and you can terminate the oscillations. So it's quite a neat explanation for a number of different things. So the conclusions from that are that our first assumption was actually plausible but very damaging to the system. So we got something that could oscillate, but just neglecting the buffers left us with a very rigid system. And if you add the buffers in, you can explain these different things. So now on to the main topic of the talk, the spatial modeling. So this is the supplemental work on the web of a very, very nice paper that came out 2 years ago by David Barker's group. And they developed a nuclear-specific chameleon line. So that's a nuclear-specific dye that will only, this nucleated nucleus, that will only go on in the nucleus when you look at the oscillations. So whereas before there was called perinuclear oscillations because you couldn't distinguish really where they were, these are clearly happening in the nucleus. So oscillations within the nucleus, and you can see that they're not homogeneous, so there's some spatial patterning to them. And there's a whole number of images in the main paper that I haven't put in because they never come out well on projections, where you can see that it's a very interesting system. It's not just on, off with spikes. There's lots of spatial patterning going on within the nucleus. We wanted to try and understand that because going back to Jörg's talk, the spatial signatures, they won't just be spatially integrated oscillations, the kind of thing we were looking at previously, they're going to be local. Those are the things that the proteins are going to be challenged with when they have to decode something. So the spatial aspect of this could be very important for determining specificity. So again, we can set up a very simple model. So cytosol looks just like a root hair to me. Nucleus, and we're going to put in a few elements. So because of this hypothesis of free diffusion through the pore, we're just going to see if we can try and get oscillations going on the outside, and see if they're going to get transferred to the inside. And that's a hypothesis that's been mentioned in many papers, that the nuclear oscillations are actually, in fact, just oscillations on the outside diffusing through to the inside of the nucleus. So this is actually rather complicated modeling. It's taken us quite a while to get to where we are. It's 3D reaction diffusion modeling. It may look simple to you, but this is quite a complex geometry to model. We're going to be looking at this system inside and outside the nucleus and free diffusion through the nuclear pores. So there are estimates of how many pores there are in plant cells, estimates of the density, and from that you can work out roughly how many there are. So we've scattered 500 pores on nucleus, diameter of 9 nanometers, and we just couldn't get that working. We can get nice spikes arising, oscillations around the nucleus. We saw nothing on the inside. Before I get to that, I'll just show you a bit more of how we did that. So these are just some equations I stole off the web to try and impress you with. Now, actually, this is what we've done. So you can set up a reaction diffusion equation for the system. I'll just go through briefly what they are. The first term is simply the diffusion of its G, for reasons I'll get to soon. That's the calcium concentration. So diffusion, then we have a pumping out term, and then we have a source term, which is from the calcium channels. And we can spread as many channels over the nucleus as we want. We just have to have a summation of all these different sources. And we're lucky enough that this equation actually has a solution to it, an analytical solution. And that's this second line here. You can expand or develop a solution of this equation in so-called Green's functions. And horrendous as this equation might look to you, it's perfect for programming. All the components of this, these are rather hideous special functions, Bessel functions, Legendre polynomials. But we know how to code those up. So we can code those up, and then you have an analytical solution for the calcium concentration everywhere in the cell. So you're avoiding numerical integration and all the associated problems with boundary value problems for solving PDEs. And there are a few constraints of that. You have to solve this root equation here to make sure the flux doesn't go through the boundary. But those are all things you can get by. So that's a very neat, and that makes it a very fast way of solving the equations. As I was saying a slide earlier, we couldn't get oscillations on the outside to go to the inside. And we even went to quite some extremes there. So we threw in 4,000 nuclear pores, opened up the diameter of the pores to what's thought to be one of the maximum rate, about 25 nanometers. And we even turned the pumps off just to make sure the level was continuously going up. Within the time frame of 10 seconds, and that's roughly, so the oscillations are thought to be delayed between inside and outside, depending on the stimulus, but reports of 1 second to about 7 seconds. We couldn't get oscillations on the inside. So that's suggesting to us that the machinery has to be on both sides. And even on both sides, with these large parameters, we can very nicely start a wave generation on the outside of the nucleus. We couldn't really kickstart it in the center. So that's also telling us something about the mechanism. So if calcium itself can't induce these oscillations, a purely ligand-gated channels, unlikely that will have the same problems. So we think there must be some voltage sensitivity there to get the synchronization working. So that's interesting too. So what we thought was kind of an obvious thing of diffusing through is not at all the case. These two compartments on the timescales we're looking at can be not in equilibrium despite this free diffusion through. So we don't know if there is free diffusion through the nuclear pore. papers saying that there are oscillations independent, autonomous within the nucleus and outside. Many of them suggest that, yes, well, you can block the nuclear pores, or the nuclear pores act like channels that can be gated and closed. And that may well be the case. But what this has shown is that even if there was free diffusion, you can still get autonomous oscillations inside the nucleus and outside the nucleus. And it also suggests to us that the machinery that will give rise to these oscillations has to be on both sides. So that made us look a little bit harder at where these components might be located. And this was a pump that was predicted by a bioinformatics analysis, and what you probably can't see at the very top slide is a GFP fusion with this pump that puts it at least in the nuclear region, but you can't see much more than that. So then we went to immunogold labeling, and what you probably can't see as well, but you can take my word for it, that's these dots, and we've just counted the dots on the inside and the outside of the nuclear membrane, and they're clearly on both. And likewise for the ion channel. So of course we're missing the actual calcium channel, the most important thing, but people are looking into it. So I'll just summarize where we are on that. So buffers may play a key role in initiating, shaping, and terminating the calcium signals. We've shown, at least in theory, that they can, and that offers a nice explanation for what might be going on in terms of the signal transduction from nod and mixed factor perception to kickstarting these oscillations in the nuclei. Oscillations clearly occur in the nucleus as well. That's been shown, but now by many groups. And then membranes play a key role in regulating these oscillations. And the nuclear pores may well allow the free diffusion of calcium, but at least in our hands, we couldn't get them to actually transmit oscillations. And that's suggesting that the machinery has to be on both sides of the nuclear membrane, and that, of course, allows you to have autonomous oscillations on either side and specific regulation. And I mean, just to bring the point home again, that implies that we have to understand the spatial temporal dynamics within the nucleus to understand the decoding side of things. CCAM-K, the protein that's thought to decode these oscillations, isn't going to first do a nice spatial integration of the nucleus and then start decoding. It's going to be exposed to these local signals, and the initial modeling we've done on this would suggest that the calcium signals around the membrane are quite different from the ones that occur through interference in the center. So that's a nice analogy to Jörg's talk, where he was saying that these decoding proteins that locate to the membrane. Could you imagine something actually similar within the nucleus happening, that they're going to specific locations to read out? So we're working on that. So with that, I'll just say what the next steps are. So today I was telling you about what we've done on the encoding side. We've done a lot of work on trying to discriminate and actually analyze these different signals. And of course, we're working a lot on decoding and have something that we think actually does do frequency decoding now. So this will all feed into a model for the whole process. We're also working on cell wall remodeling, and can we understand the whole process or reproduce the whole thing in silico? So we want to get to the equations of symbiosis. This is a rather crude framework still, but we're filling in more of the details as we go. So with that, I'll finish. I talked mainly about Darren and Saul's work today. Saul developed this first model of calcium signaling with help from Crassi from Bristol, and Darren's done this very nice work on spatial temporal modeling and diffusion through the nuclear pores. And I'm very grateful for Giles Aldroyd and Alan Downey. They're the biological brains behind all of this work. And if anyone's interested in more in calcium, I can recommend the book by Graham Chapman, or just ask the Calcium Kid. Thank you.